Sam Allan is a Master's Research Candidate with the Fish Ecology Lab in the School of Life Sciences at UTS and an OFF research grant recipient. His presentation will focus on research conducted over the past 18 months in Sydney, concentrated in Botany Bay, Bondi and Manly, as well as Port Stephens. Sam is passionate about marine conservation and is actively involved in local diving and surfing communities, as well as the marine science research field. He believes that conservation efforts for unique species such as sea dragons can be effective in garnering awareness for broader conservation issues. So with that brief introduction to Sam, I'll now hand over to you, Sam, to give us your presentation.
Great, thank you, Kim. Thanks for having me on, everyone, and thanks for supporting me with the grant when I received it about a year ago. That was really great, and it made the research just that little bit easier, which is good. So I'll just share my screen now. How's that? Can you guys all see that?
Yep, that's good.
Yeah.
Alrighty. Okay, well, I'll get started. So just as Kim just mentioned, today I'm going to be presenting on my research that I've been conducting over the past 18 months as a part of my Master's by Research degree. So I'm due to complete that by the end of the year, planning to hopefully submit before Christmas. And my presentation today will focus on the research that we've done, which is detailing the range limits, particularly focusing on the northern range limits, and then looking at the habitat and prey associations of the weedy sea dragon. Phyllopteryx taeniolatus is the scientific name. And just I'd like to acknowledge at the bottom there, they are my collaborators who've been helping on the project. So David Booth, Summer Clanton and David Harasti have been my supervisors throughout this project, and Max O'Connell is another master's student who has been instrumental in getting all this research completed. And just before I get started, Kim already did it, but I'd just like to acknowledge the Bigtigal people of the Eora Nation, because most of this work was conducted in Cornell.
Mm-hmm.
Okay, so just a little bit about myself. As Kim mentioned, I'm really passionate about marine conservation. So I completed a Bachelor of Environmental Biology in 2018, and then pretty much led me straight into starting this Master's, which is focused primarily on marine conservation and sea dragons. And throughout this process of studying sea dragons, I've been able to collaborate on other projects as well, where we've been monitoring endangered seahorse species or monitoring tropical vagrant fish that come down from the Great Barrier Reef onto some established coral beds that are found around Sydney. Okay, all right, there we go. So not sure how much anyone knows about sea dragons, but sea dragons are members of the family Cygnathidae. So that is a family of fish that comprises seahorses, pipefish, pipe horses, and sea dragons. So sea dragons are pretty unique, charismatic Australian species, and they're all endemic to Australia as well, particularly to the temperate reefs of southern Australia. They're cold water species. So the three species of sea dragon are the weedy sea dragon, which is on the left there. You've got the leafy sea dragon, which is on the right. It's fairly ornate, very unique looking, and that one's only found down in South Australia and around in Victoria and a little bit in Western Australia too. And then this middle one that I've got there is the ruby sea dragon. This one's really interesting because it was only discovered in 2015. So these other two sea dragons, we've known about them since about the sort of mid 1800s, but the ruby sea dragon, lives in much deeper water as well. So they think the reason that it's red is because at the depths of about 50 meters where you find it, red light is the first light to go underwater. So the theory is that it's fairly difficult to see when it is underwater and camouflages really well. So my research is just focused on weedy sea dragons. And then this map here on the right just shows you their distribution throughout southern Australia. So of note on this map is the northern limits of where they occur. So you can see just over to the right here is Port Stephens. And then over on the left is Geraldton in Western Australia. And then, so they are the reported northern limits of where you find sea dragons. And all these navy blue dots throughout the rest of Australia are just verified occurrence records from the museum of where you actually find sea dragons. So what's the IUCN Red List, which is like the threatened species network, basically, they list the Weedy Seadragon as least concern. However, prior to this, as recently as 2017, it was considered a near threatened species, but it was reassessed and decided to be least concerned. However, a study that was done in our lab in 2011 showed that there was population declines in sea dragons in both Sydney and Tasmania. So just a little bit more background on sea dragons as well. One of the main traits of signated fishes, sea dragons, seahorses, what makes them so unique is the fact that the males are the ones that get pregnant. So the male brood the young. And as you can see over on the left here, I've just got a few, basically the different life stages of a sea dragon. So you've got up in the top there a pregnant male. You can see it's got a full brood of eggs attached to the underside of its tail. They carry up to about 300 eggs at a time. But they do have a very high juvenile mortality, upwards of 90%. And the juvenile dispersion is very low as well. So once they do hatch, they don't tend to roam very far from where they're born. On the bottom left, there's just a new baby, very, very young sea dragon, probably only a couple of days. When they hatch from their egg, they basically come out as perfectly formed miniature sea dragons. And then just on the right, there is a sub-adult. So it's not quite a fully grown sea dragon, but you can see the body size difference between that picture and the one up above it. So despite being such a unique and charismatic species, sea dragons are relatively understudied. The majority of the work, at least on the ecology of sea dragons, detailing the habitat and life history, has come from another student who was working with our lab in the early 2000s. So they have really small home ranges, as I mentioned on the previous slide. Basically, wherever you find them, they're not going to go very far. They have very highly structured genetic diversity between, there's a recent paper come out that shows from Sydney to Victoria, there's actually really, really strong genetic differences in sea dragons, almost to the point of being considered a subspecies. They look different, they occupy different habitat, they're found at different depths, so the differences, and as well, they're different genetically. The only thing that we had known previous to the research which I'll present throughout this paper, throughout this PowerPoint, was that sea dragons are found at the kelp sand interface. So basically, particularly at Cornell, if you go diving, there's this hard line of where the kelp beds finish and where the sand begins, and then it's just the middle of Botany Bay pretty much. Along that line is where we expect to find sea dragons the most often, because it's supposedly the best trade-off between where they can still camouflage amongst the seaweed and where they can eat their main prey, which is mysid shrimp. So mysid shrimp are small little crustaceans, look like mini prawns, and they form big swarms. And basically, the sea dragons with those really long snouts that you can see, they're suited to feeding on these really highly motile prey. So basically, they suck in the mysid shrimp. So a really fast motion, like a snapping motion, and it draws up the mysids, and then they can eat them. And they don't have many fat reserves either. So an interesting sea dragon fact is they pretty much constantly have to eat. And then another unique thing about sea dragons is you can identify each individual sea dragon just by the spot pattern on the side of it. So every single one is unique, much like a leopard or like a fingerprint on a human or something. Each one is unique. And you can also tell their sex just by looking at them as well. So this image on the top here, as you can see, it's got a much deeper body. And this here, the colca, where the tail joins to the body, is almost a right angle. Where this is a male, the body comes up a lot more. And this is a much more, almost like a 45-degree angle. So that, unless there's eggs on the tail, that's probably the easiest way to tell, but not always the case. So that's just a bit of info on how to ID a sea dragon. So this is just an example of using identification software it's about 98% effective in IDing individual sea dragons. This was just a photo in July on the left in September of last year, where I found the same sea dragon and was able to ID and just know that it was still the same one. I think this particular one, we saw about 7 times over the course of the study. I think the most I saw one was 10 individual sightings. And this just here, this is of all those spots, basically what you do is you hone into one little section where you see it come up from the ventral fin here, and it's 2 of these spots along the lateral line backwards. And then you circle these points with the program, and then it will basically spit you out about 10 different sea dragon options from your database that you've established. And then you can determine whether or not it's a correct match. So just a little video here. This is just showing some sea dragons, actually in situ at Cornell, and it's a little bit difficult to see, but just down over here, there's a big swarm of mysid shrimp, which is what they eat. But those 2, it's not often that you kind of see them paired up like that, so they might potentially going to be doing a mating dance or something and might have scared them off, not sure. But they have a pretty unique mating ritual, which hasn't been recorded very often. So this is just a video of me basically getting those photos that you need to identify the individual sea dragon. So what you need to do is make sure that you have a photo from both flanks of the sea dragon. If you've only got a photo of one side, it might be difficult to, if that's really similar to another one and the other side is different, it might be difficult to determine A, if it's a new sea dragon, or B, if it's one that you've seen before. So I've had plenty of sea dragon photos to sift through over the past 18 months and got fairly good at IDing them. I think over the course of the last 18 months, we had 62 sea dragons across all of our sites and saw them about 120 times, which is almost twice per dragon. But so a bit more, that was just a bit of background on sea dragons in general, but just to give you a bit more direct information on the research that I've been doing, which you guys were nice enough to fund. Thank you very much. So primarily we had 3 aims, and it was kind of broken up into 3 papers or 3 chapters, which will go into my thesis. So the first of our aim was to determine sea dragon habitat preferences. So I mentioned before that sea dragons are found at the kelp sand interface, but that is, up until the research that I've been doing, that's all we knew. Like, you would know that if you went for a dive, they're going to be associated with kelp. This stuff here is called Aclonia radiata. It's pretty much the most common kelp that you find throughout the temperate reefs of Australia. And we wanted to know how much sea dragons actually associated with this kelp, and did it influence their habitat selection. Secondly, what we were hoping to do was then apply any of this information that we developed in the first phase of this study, looking at sea dragon habitat associations, to see if you could predict where you actually find a sea dragon within a site. And this is significant because if you can then do this, it opens up a lot of avenues for developing conservation strategies and things like that. So if you can look at an area and be like, okay, there is a certain amount of habitat or mysid shrimp, or it's a certain depth or whatever, it makes it a lot easier to conserve those areas of habitat, which will then benefit the sea dragons. And thirdly, what we were hoping to achieve was to properly assess the northern distribution of sea dragons in New South Wales and Western Australia as well. I'll go into that in a little bit more detail further through the presentation. But to date, there hasn't really been any sort of large-scale assessment of sea dragon range limits. So, as I said, we were hoping to amend that, see if we could get some more info on sea dragon range. So that leads me to the first phase of the study, and this was just recently published in the Journal of Fish Biology. So if anyone is interested in reading it, I can send you a full copy if you like, because you'll probably hit a paywall otherwise. But if you feel free to contact me and I can pass it on. This one was titled "Space Use by Weedy Seadragons, Looking at the Influence of Habitat and Prey." So as I mentioned a moment ago, sea dragon habitat preferences are currently understudied. basically all we know is that they were found on the kelp scene interface. And what we were hoping to do was work out how much these sea dragons actually associate with the kelp habitat. I don't know if you guys have been noticing, but every time I've put up a sea dragon photo, more often than not, it's in around this Eclonia kelp that I was talking about before. So we were hoping to look, how do they associate with that kelp? How does that influence habitat selection? And also look at the interaction with the mysid shrimp. So I'll just, one second. This here is a mysid shrimp, just for anyone who's curious. Okay, so yeah, the aims: identify habitat preferences of sea dragons, and then a sort of more specific aim was to look at how kelp habitat, that Eclonia kelp, which I've been talking about, and mysid prey availability might affect sea dragon small scale distribution. So how it affects sea dragon distribution within an area. So the main study site for this pilot phase, or not pilot phase, sorry, it was the first chapter of this work, was in Cornell. So anyone who, I'm sure everyone here's spent a bit of time at Cornell. Where we would go in is, if you go in at the steps car park, and we drift down from the steps to the monument where the big whale sculpture is. And it's about it's a 210 meter study area. And from March 2020 to March 2021, which is when we're collecting data for this study, conducted 28 dives over about 20 and a half hours. We got slowed down a little bit in the middle, obviously, when COVID came up for the first time, and we had to juggle a fair bit with being able to get shore diving approved through the university. There was a lot of technicalities, but we got through it and were able to get a good amount of diving done. And the reason we selected Cornell is because it's a well-known established population of sea dragons. It's essentially one of the only locations that has actually been studied for sea dragons. And there's 4 previous papers, which were published in the early 2000s, looking at sea dragons from this spot. So we knew if we went here, we should be able to find some sea dragons. So in order to determine the habitat preferences of sea dragons, what we had to do was run out transects like this one. So this one here is a 30 meter transect. We did 2 long 200 meter transects, 210 meters, sorry, which were made up of 7 of these longer transects. They're shorter transects, sorry. And then basically what this does is helps me determine what habitat is actually present within the site. So in order to determine an animal's habitat preferences, you need to look at what habitat that animal is actually using, and then compare that to the relative habitat that's available within the whole area where you find that animal. So how we did that, as I mentioned, we have these longer transects, which is playing here, to show all the habitat present within the site. And then as we were diving throughout that site, every time we would see a sea dragon, we would run out a smaller transect, which is about 15 square meters, a 5 meter transect. And then you can record the same benthic composition, get a video of that one and record the benthic composition as well, and then compare that to what habitat is available. Whenever we also found a sea dragon, what we wanted to do was look how that would interact with mysid shrimp. So we would look to see if mysid shrimp were present in that transect or not. Okay, so this is just a few shots of how we actually determine the benthic composition. So there's a program, this image on the left here, and what it does is it imports random points over an image. And this image, I would then go through and I have to assign what is underneath those points. And then that gives you an estimation of what's present within the site. So all told, it was about 30,000 points of assigning habitat. So it took a little while, but it gives you a good idea of what's present. And then, so we would do that for areas where you would find sea dragons, areas where you would not find sea dragons, and then also for the entire site to see what was there. This is, I won't go into too much detail on this, but this basically just shows the main habitats that were present. So sand, rock, and Eclonia, which is that seaweed kelp that I've been talking about, are the main habitats that you find at Cornell. So the kelp makes up about 20% of the overall habitat. Rock habitat makes up about 31%, and just bare sand is the dominant habitat, and that's about 47% all told. So once we had done all these dives and found all the data, we determined that sea dragons have a strong preference and a strong association for this Eclonia habitat. So over here, this graph, this line is representative of a displayed preference for a habitat. So if these error bars here fall above this line, then an animal significantly prefers and associates with that habitat. If it falls below the line, then they'll avoid that habitat. And if it's within, like sand here, then there's no association. So what we showed was areas that are dominated by Eclonia sea dragons are more likely to be found there. So when you look at all the habitat that was present within the site, there's a lot of sand and there's more rock than there is kelp. But what the sea dragons are doing is actively seeking out those areas of higher kelp so that they can camouflage and feed. So what we then found out was this graph basically just shows that these white bars here are the distribution of kelp throughout the site. So up towards this higher end, you can see there is very, very few areas where there's really high kelp cover, but there's a lot of sea dragons found in these areas, which means that throughout the entire site, they really are actively seeking out these areas of between 40 to 80% cover of kelp. That's what they like. So additionally to seeking out these areas of higher kelp cover, what we also showed was that across all habitat types, so Eclonia, sand and rock, that sea dragons would look for areas where there was also mysid shrimp present. So what these grey bars represent is the proportion of time that sea dragons were found with a mysid shrimp swarm around them. So across all habitats, it's at least almost 80% every single time they're found associating with this food. So basically tells you that whenever you're looking for a sea dragon, if you look for areas where you find mysid shrimp, and if you look for areas where you have a bit more kelp, upwards of 40%, then you're probably much more likely to find a sea dragon. So just to summarize the findings from this part of the research, as I mentioned, sea dragons specifically prefer areas of, between 40 and 80% of Eclonia cover. They strongly associate with mysid shrimp. And then basically this led us to ask the question of how can we use these results to then better understand sea dragon distribution, and can we use these results to help maybe predict where we might be able to find a sea dragon within a site. Which leads me to the next chapter, or the next paper of my research. This hasn't been published yet, but it is hopefully going to be submitted this week to a journal, and then we can, hopefully it'll get out before Christmas, but we'll see. So a little bit more background for this phase of the research. So what I was hoping to do was apply the knowledge developed from the previous chapter that sea dragons prefer the kelp and the mysids, and see how this could be applied to several sites which were stretching from Sydney to Port Stephens. So as I mentioned at the start of the presentation, Port Stephens is the northernmost reported limit of sea dragons on the east coast of Australia. And this is the first study which has assessed sea dragons north of Kurnell. So outside of Sydney, but as I mentioned, most work has been done at Kurnell prior to this. So what we wanted to do was test if the knowledge that we developed in the first chapter of this research could be applied to determine if sea dragon occurrence can be predicted within a site across 200 kilometres of coastline, Sydney to Port Stephens, and also to have a look and assess the relationship of mysid density within an area to sea dragon density within an area. So our study sites for this phase of the research were Kurnell, which I had discussed before. And then we also had North Bondi. So North Bondi was a site because that was included in those other 4 papers that got published in the early 2000s. Cabbage Tree Bay in Manly this is the first time that population of sea dragons has been studied. And Broughton Island, this is also the first time that population has been studied. So We conducted 47 dives over almost 36 hours. And just of note as well about these sites is, as I mentioned, that sea dragons like to be found on that kelp sand interface. It's at Cabbage Tree and Broughton Island. There's not really a kelp sand interface. It's just like a big 3D area of kelp, and the sea dragons are kind of found within it. Whereas at Cornell and North Bondi, there's a hard line of where the sand meets the kelp, and that tends to be where they congregate. This is just a few action shots of where we were. Unfortunately, this day on the left, we went all the way to Port Stephens, and it was just after the floods in March, and we got in the water and you couldn't see your hand in front of your face. So we just had to get out of the boat and then turn around and come back. This was a bit more of a successful trip. That's where we would dive at Broughton Island for the sea dragons. Really, really nice spot. Just some nice photos. That's the reason they're there. So what we wanted to do was look and compare the habitat composition across all these sites. So we did this in the same way that I had a video of earlier, where we would run out transects within a site to determine whether or not they were habitat composition was the same. And then, as I've mentioned, we wanted to develop a predictive model of where you can find sea dragons within a site. So we did this by taking measurements from areas where you would find sea dragons and areas where you do not find sea dragons, and then basically comparing those two to see if we can develop a model that will help us predict where they can be found. So this graph here just shows the habitat composition across all of the sites. So along the bottom there, you've got going from left to right, Cornell, North Bondi, Cabbage Tree Bay, and Broughton Island. And what we showed here is that the habitat did not differ across any of the sites. So despite the fact the sites are about 200 kilometers apart, the habitat at each of these spots is the same. So this could be useful for further research when you might be looking for other areas where you can find sea dragons. You might have a rough idea of what sort of habitat you're going to be looking for. So what we have here is we were assessing the relationship between the sea dragons and the mysid shrimp. And what was interesting with this is as the density of mysid shrimp within a site would increase, then so would the sea dragons. So what we were hoping, or what we think this could possibly be used for, is the main reasons or the limiting factors of sea dragon distribution aren't really well known. So as their northern limit is Port Stephens, we're not exactly sure why they can't go any further than that. We think it could be related to, well, we thought it could be related to the kelp cover, but there is a little bit of kelp mapping out there, and there's some predictive modeling and things like that show that around Port Stephens, the percent cover of kelp is actually quite similar to that in Sydney. So possibly not that, but this relationship of mysid shrimp might be a strong factor in limiting sea dragon distribution. So if you were to go and look at some sites further north of Port Stephens, of Broughton Island, and there was lower densities of mysid shrimp, then that might be directly correlated with the sea dragon densities as well. Okay, so yeah, we saw during this phase of the research, we spotted 114 sea dragons, 62 sea dragons 114 times from March to September 2020, 2021. And what we were able to do was, using that knowledge that we developed from the first thing, is that we were able to predict, based off a clonia cover, if there was a higher level of aclonia, that 40 to 80% range, and the presence of mysid shrimp, then you can, at a rate of 71.2%, you can accurately predict where a sea dragon is going to be found within a site, which was a fairly interesting outcome of the study, we thought. And hopefully we're going to get it published soon, fingers crossed. So just a summary of this study. As I mentioned, this was the first time that a population of sea dragons has been studied at Broughton Island. It's also, interestingly, the first time that a population's even been recorded at Broughton Island. So we kind of had a rough idea that they had been there, but there hasn't been any actual research or anything like that put out that actually documents a population of these animals at this location. We showed that increasing Aclonia cover and the presence of mysid shrimp can be used to predict where a sea dragon might be found within a site. And what we're hoping with this is that, as our study was only from Sydney to Port Stephens, that the findings from this could maybe be used and further developed to help predict where you might find sea dragons throughout their entire range all the way across into Western Australia. Obviously, that would require some pretty extensive surveying, but I think this gives a good foundation for helping to better understand how these animals associate with habitat all the way through their distribution. We hope that this can help prioritise areas of Aclonia for conservation as well. So Aclonia kelp habitat is fairly abundant in Australia, but there have been noted declines in a number of areas due to increased numbers of sea urchins, which graze on Aclonia, and also just to increased temperature. So if the East Australian current is continually getting warmer, then it's going to negatively affect the distribution of kelp, which would then in turn negatively affect the sea dragons, because if they don't have the kelp to hide in, then it's going to be a lot more difficult for them to survive, because they're pretty poor swimmers, and without their camouflage, they don't have a lot else going on for them. So this is just some brief information on the other phase of the research where we're looking at the distribution of sea dragons. So this wasn't as much a field component as the other two parts of this study were, more so a review of the available sources on distribution and things like that. So we found that there's been a fair bit of variability with the actual reports of the northern limits of sea dragons. As I mentioned, so that we were the first, this is the first study to record a population of sea dragons on Broughton Island. So this map here on the left is in Western Australia, and the one on the right is New South Wales. And basically, each of those points is just from some fairly important sources. That just shows you where the northernmost reported sighting is. So on the West Coast, there's about 400 kilometre disparity between different sources. One we've got there at the bottom is RedMap. The one on the top is Dragon Search, which was a citizen science project that ran from 95 to 2005, just of divers recording where they would see sea dragons. And the northernmost one was the Abrolhos Islands. And the southernmost report from RedMap, which is another citizen science-based website, is about 400 kilometers south. So this variability really just highlights that we don't know that much about sea dragons, and particularly if you want to help protect them, and if we are going to develop conservation strategies that are actually going to help them, we have to know where their population stops and why they can't establish populations further north than that. Because on both the east and the west coast of Australia, you have those strong oceanic currents, the Lewin Current on the west coast, the EAC on the east coast, pushing down warmer water further and further into the southern parts of Australia. So if these locations further north get too warm, if it's warmth that's stopping the sea dragons or whatever that might be, it could have really, really drastic implications for them as climate change continues to worsen. So basically what we wanted to highlight here is that there needs to be further work done. There needs to be further surveys done, particularly on the West Coast, to better understand the distribution of these animals. So just a bit of an overall summary on the project findings. So the main finding that we address throughout here, I've mentioned it plenty of times already, but is that we showed sea dragons strongly associate with Aclonia habitat. And previously no one had showed this, it hadn't been done. And we also showed that they associate with the mysid prey. We use these two factors, the Aclonia and the mysid prey, to predict where sea dragons might be found within a site. And as I mentioned, we also provide the first confirmed sighting of a population of these animals at Broughton Island. So some of the future directions which we're looking into at the moment is, so at the start I mentioned that sea dragons have really highly structured genetic populations. So this is particularly evident between New South Wales and Victoria. So these images here on the left is a sea dragon found at Cornell, and you can see lots and lots of that Aclonia habitat that I've been talking about. However, interestingly, this sea dragon on the right is from Flinders Pier in the Mornington Peninsula down in Victoria. So as you can see, this habitat here is very, very different. So this is a seaweed. I believe it's Cystophora, and then there's a lot of seagrass habitat that they associate with. So what's really interesting for these more southern populations is it appears that they actually don't associate with this Aclonia habitat, which kind of just adds more fuel to the fire of the highly structured genetic diversity, possible subspecies complex. If they're associating with different habitat in a different spot, they look different, they're found at different depths, their genetics are different, then they're pretty different. Another really interesting thing is you can see that this sea dragon on the right, it's a lot shallower. The typical depth that I've been finding them is between about 12 to 21 meters, whereas these ones on the right that you see in Victoria, you can see them snorkeling. They're only at 2 or 3 meters depth. So that could be potentially related to depth, something like that. Again, a really important future direction to take this research in is to go and assess sea dragons throughout their entire range. We need to look at populations in Victoria. We need to look at more populations in Western Australia. And it would be nice just to look at some down in South Australia too. But we can, that's where we're hoping it's going to go from here. But yeah, there's definitely a lot more work to be done. And just if you're interested, just a few of the research outputs that we've had so far. So I presented this research at the Australian Marine Sciences conference in 2021, which was in, I think, July. And then also presented it at a Signathid conference, Signathid Specialty Conference, which was held virtually in China. I was supposed to be going, but obviously COVID meant I didn't. But it was still really awesome to be able to present and just talk to other researchers from around the world who've been researching Signathids, get their opinion on what I was doing. And luckily I was awarded best student presentation for that, which was pretty nice. Research papers, the first paper that I presented in this talk, as I said, has been published. So please, if you're interested in reading it, let me know. I'd be more than happy to send you out a copy. The second chapter and the third chapter, looking at their range, we're still getting written up, but they're pretty close to being submitted. So I hopefully going to be able to get those through without too much trouble, but we'll see. I'd just like to acknowledge a few of the people who helped me along the way. They're all written there. And then lastly, acknowledge all of you guys. It was really an amazing help to receive this grant, and it was really, really great. Helped the research a lot, particularly with our trips all through Kurnell, getting the dive gear maintained and filled. And then particularly when we were going up to Port Stephens, we had to go out probably like 15, 20 kilometers off coast. So being able to like get a boat, get a skipper, everything like that, really couldn't have completed the research without it. So thank you everyone who was involved. And thanks for listening, and happy to field any questions that you guys might have.
Well, thanks, Sam. It was fascinating to learn about the weedies. They're really interesting.
They are, aren't they? I love this video. Just animals. The two of them together. It's yeah,
it's good stuff. Yeah, so there's some questions on the chat line. Where's it gone?
Oh, sorry.
Must have gone to the right. Oh, you've stopped sharing.
Yep. There we go. Yep, it's back up. Sorry.
Okay. And are you able to see those questions?
My question was did you run the transect along the sand line or just at a particular depth and direction, or did you actually just follow the sand line for the Kurnell study?
Yeah. A bit of both. So in order to get an idea of the habitat that was available within the whole site, we had to sample. Yeah, we did one that went right along the sand line, and then we did one that was 15 meters parallel of the reef, and did that 210 meters as well. And that gave us an idea. That's the whole area that we would search.
So both 210?
Yeah, so 420 all up.
Okay. And I didn't quite understand, when you saw a weedy, you said you did 15 square meters. Do you mean you went out 3 by 5, or what did you mean?
Yeah, so at every weedy sighting, at the point of initial sighting, I would drop the transect tape and then run out a 5 meter tape. And then as you're swimming along that 5 meter tape, it's 1.5 meters either side.
So still parallel,
3, 15.
not swim out. You didn't go perpendicular. You'd meant you'd do 5 meters at that. Okay.
Yeah, exactly. Yeah.
Okay, you've asked us to ignore your last question.
Well, the third question, then I saw the
Lou?
title said number of swarms. 200, because I was like, how in hell would you count mice?
Yeah, big swarms of them.
Okay, the whole swarm.
Yeah, the whole swarm. Yeah.
Better eyesight than I do. Okay.
Yeah, it would be hard. You get your eye in for them for sure, but they are pretty shifty.
Yeah.
And then we've got one from Martin Puchett. Could there be a lower abundance of mysids further north? Could that explain why weedies don't go further north?
Yeah, potentially. It's really something that hasn't been looked at all. We did originally have aspirations to get up there and have a look, but there's been so many obstacles over the past sort of 18 months. Like I started pretty much when the bushfires did, so that shut off our access going north, and then obviously lockdowns, the floods earlier in the year really kind of ruined the visibility for about 2 or 3 months. So I think definitely it could possibly be a factor. If there is less mysids there, then I think it's definitely, the two are definitely associated, but no one's actually been up there to look at the mysids, so it's hard to say.
Okay. Does that one need answering, Lou? Are mysids a single species?
Many species.
Google says, yeah. Google says, I just wondered if do they eat, I've just always seen them as just a big amorphous kind of.
Yeah.
Would they eat only certain types, do you think?
No, I've observed them eating. So if you can spot the mysids when you're diving, there's some that form a bigger ball shape. They have a really clear body with like black lines through them. Something I'm not too sure how to speciate them. They do look quite similar, but I've seen them eating different ones. Definitely different species. Yeah.
Okay. Here's from Matt. What is the main predator preying on dragons?
There's no known predators actually, which some things do eat them, because quite often you'll see a sea dragon that might be missing a few of its leafy appendages or something like that, but there hasn't been anyone observe what actually preys on them. So I was diving last week and I saw one that was actually missing all of its tail, like it had been freshly bitten off, but really have no idea what it could be. There's certain things that it's like octopus, certain types of leather jacket, cuttlefish, they eat seahorses, but seahorses are a lot smaller. So maybe them, but yeah, really, again, supposedly no known predators.
More research needed.
Definitely.
Humans are sea urchins overabundant, and what are their main predators?
Yeah, so the big problem with sea urchins is particularly down in Tasmania. So they used to have all these like really well established like giant kelp forests and things like that. And some of the main predators on sea urchins are within Tasmania. You get the eastern rock lobster. So a big problem there is that lobsters are overfished, and then you get more sea urchins that aren't getting eaten by the lobsters, which then the sea urchins eat the kelp. Around Sydney, sea urchins, I wouldn't say are like overabundant to the extent that they are down in Tasmania, but there is definitely a problem with increasing abundance and reducing. It is negatively associated with kelp abundance. So the main predators around Sydney, blue gropers love to munch on urchins. If you get like a really tamed one on a dive and you like bang your hands together, they think you've got an urchin, and they'll come over and you can just swim around with them, which is fun. But yeah, gropers are the biggest thing around here that would eat them.
Okay. Graham Lauscher, can you see anybody with a hand up as opposed to putting a message on the chat line? You're muted.
No, I can't see any here.
Okay, I just thought there might be some people using that method, but no, so we'll continue on with these ones in the chat. Big fish that are overfished.
Do numbers vary during the year? I think that one.
Yeah.
Yeah.
Are they affected by seasonal temperatures?
Not so much, because they're such poor swimmers. really get affected by it. The breeding season is from about June right up sort of till the end of the year, even into like January, February, because the water is the warmest in sort of March to, February, March is when the water's warmest. So that's when the babies tend to hatch. But overall, their abundance doesn't really seem to get affected by seasonality at all.
How long is their average life?
Average life, another good one. They've had them live in captivity, so they're a really popular animal to have in captivity. And at aquariums and stuff, they're fully protected, so you can't just have one in your fish tank. But aquariums and things like that do have them, and they've been noted to live up to 9 years in captivity. In the wild, it's a bit more difficult because obviously you've got to be diving all the time to tag them or take the photos of them and ID them. But they've seen them live up to 6 years in the wild at Cornell. So sort of 5 to 9 years, roughly.
One from Jeff Innes. Do you have any idea of spatial range of individuals? Do they stay in the one area or do they roam?
Yeah, so their home range is about 100 meters, give or take. The furthest one that has been documented, at least in a research paper, there was one that was sighted at Cornell, and then it was sighted at Bear Island. Probably that would be to do with a lot of currents or some a big swell that would push it over that far, because typically they would only roam up to 100 meters within a site. And the juveniles, they have an even smaller home range. So very highly site attached.
And would the tail regrow if bitten off?
It would not. But interesting, there's been people have seen and got photos of a sea dragon that's had a male sea dragon, had most of its tail bitten off, and then it still had a brood of eggs, which was pretty cool. Not a full, obviously, I guess wasn't a full brood, but still pretty neat to see. It's missing half its tail and it's still got a lot of babies.
Yeah. Are we down to the end of the questions so far? Anybody out there wants to just pop in a question?
How does male brooding work? Just came through.
Oh, there. Yep.
Yeah. So basically, again, this is a bit of a tricky one. It's never been recorded or observed how this happens. But what they think is, well, the females have the eggs, and then somehow when the males know that they're going to be getting the eggs, the tail gets a lot wider. It gets really, really spongy, and then some way or another, again, this hasn't been observed, but then the eggs are deposited onto the tail, and you can see, even after the eggs have fallen off and the little babies have hatched, you can see the tail's still really wide, and where there's the little sort of imprints as to where the sea dragon babies used to be. But yeah, no one, as I said, no one has seen it, and it would be a really, really interesting video to have.
Can I ask a question, Kim? It's Mari.
Yes, Mari.
What's the purpose of them? How do they help in the environment, in the ecology? What do they do and achieve, and how do they add to everything?
Yeah, it's a good question. So the main thing that they do is so they're not, like I mentioned about the blue gropers before, controlling urchin numbers or something like that. Sea dragons aren't really considered to be a keystone species or anything like that. But the main thing, particularly with charismatic species like a sea dragon, like a seahorse, is that their biggest role is mostly for, well, one thing, at least from a anthropomorphized point of view, is that they can be flagship species for conservation. So looking at a unique animal like that really tends to get people really engaged with being if we help protect this kelp's a little bit more boring than a sea dragon, so people might not get as involved. But if you can bring it back to something like a seahorse, bring it back to a sea dragon, then it can really help garner conservation awareness. It's much like melting sea ice and polar bears losing habitat, or giant pandas and bamboo forest destruction, things like that. But in terms of the actual ecology, not a great deal.
Oh, thank you.
That's right.
Just one above there. Is the range of seahorses similar to sea dragons?
Smaller. Yeah, so seahorses have what's called a prehensile tail. So they'll latch onto things and float to a seahorse home range is, only a couple of meters. Whereas because sea dragons are more free swimming than a seahorse, they just float around in the kelp. They can be upwards of 100 meters. So smaller, but still they're both quite small in terms of fish anyway.
Lou, you've written controlling mice at numbers.
Oh, that was a joke.
Yeah.
That
was funny.
I'm assuming that wasn't I'm assuming that wasn't true.
I liked it.
Okay.
I wondered, you can't answer, but I didn't realise the ones down south were so uncamouflaged and so in such shallow water. I was quite surprised. And I suppose they don't have predators, so there's no reason you would have to, but I just never imagined they would occur not in something like a colonia.
Yeah, it's really kind of bizarre because the coloration of it is so well suited to a colonia. But you get down. I haven't dived there. It would be really interesting to see what they are, but they're a lot smaller. It's a lot shallower, and they just found over seagrass and things like that. So anyone who dives with sea dragons in Melbourne would, exactly.
Yeah.
You can also see them if you go to Eden, Twofold Bay. You can see them snorkeling there as well. Not as far
we've
down.
been 3 times, and we got bushfired out once. We got something else out last time. Was the last holidays, COVID, I think, and then freezing cold water and an upwelling from Antarctic, so we couldn't see anything the first time. So let's not talk about the supposed ones in Twofold Bay that we've never seen.
Yeah, I was hoping to get down there in January, so hopefully I get good weather.
Yeah.
Sorry, there's one more. Is pollution or turbidity a threat?
Again, something that hasn't really been studied. There has been sort of anecdotal declines of sea dragons within Botany Bay, because obviously there's a bit of runoff and things coming out of the rivers. But probably the main threat with pollution and turbidity is that it is associated with declines in Eclonia. So if you get increased pollution, increased turbidity, it's going to reduce the Eclonia cover, which then might negatively affect the sea dragons as well. But in terms of direct effects on the sea dragons, it's not real well known at the moment.
Okay, there's one more. I think we'll make this the last one. I think I heard the genetic diversity of the Sydney population is very low. Is that right? Yeah,
really highly structured populations within an area. So there's no mixing of populations at all, really, because they don't have the capability to go to a new site. So genetic diversity is really highly structured between sites.
Okay. And we'll make this one the last one. What are the predictions for climate change?
Finish on a big one.
Yeah. You can talk for hours.
Yeah. Oh God. I guess it's similar to what I mentioned a moment ago about the impact of pollution and turbidity on Eclonia forests. It needs to be further researched. This was the first. What I've done here is the first study to show interactions with the kelp habitat. So there needs to be kind of more research done linking the predicted declines in Eclonia habitat with the sea dragons, because particularly on the west coast, those northern sea dragon sightings are right up around the range edge of where you find the Eclonia habitat. Whereas on the east coast, the northernmost sea dragons we found are Port Stephens, about 32 degrees latitude, and then Eclonia is known to go all the way up to Moreton Island. That's kind of where it stops. Whereas on the west coast, it's just north of the Abrolhos that it stops. So kind of the main goal of that study that I had looking at the range is we want to make sure that if there are populations up that far north, that we know about them, because they're so understudied that if there are established populations up there, they're going to be the first ones to feel the effects of habitat loss, because kelp, increased temperatures do negatively associate with kelp. A colonia can't really establish above, like a colonia, like I can't think of the word, like seeds, can't establish from any temperature that is higher than 26 degrees. So if those northern populations, if water temperature increases, then you're going to get significant declines in a colonia habitat, which then is probably going to have pretty significant impact on the sea dragons as well. So yeah, I guess you'll lose the kelp, you'll lose the sea dragons, I guess is the succinct answer.
Sounds like it, doesn't it?
Mm.
Okay. All right. Look, fantastic, Sam. It's really tremendous work that you're doing, and really interesting. Thank you so much. What I'd like to do now is just pass over to Melina to give you a vote of thanks from Oatley Flora and Fauna. You there, Melina?
Yep, I'm here. You can hear me?
Yep.
Mm-hmm.
Yep. Well, Sam, thank you for an excellent presentation. There were certainly also beautiful photos. You can see why there would be a flagstone species. It was really great to get an insight into how you do your research, all those hours you put into it. And as an off member, I feel proud to have supported your research, especially research that has been done for the first time. And I think we're privileged to hear the results so soon, even before going to print. So thank you very much for sharing your research. And I'm sure we'll be following your future research. Sounds like there's another PhD in it now.
Could be,
So,
haven't decided yet.
On behalf of OFF, I'd like to thank you for sharing your research. Thank you.
Thanks, Melina. Thank you. Thank you, everyone, for supporting me. It's really, really great. Thanks for having me on tonight.
Great talk. Thanks, Sam.
Thanks,
That
Sam.
was terrific. Yep. Now we're moving into our general business section of the meeting. You're welcome to hang around and listen to what we do, but
I might run off and have some dinner, I think.
quite free to go. Yep.
Yeah. Thanks everyone for having
That's
me on. I hope
fine.
you.
Yeah.
And please, if you are interested in reading the paper that got published, feel free to contact me and I can send it on too.
Okay. Yep. That'd
Okay.
be great.
Thanks, everyone.
Thanks.
Thanks, Sam. See you around.
Thanks, Sam.